Welcome back, everyone. We're going to do chapter 7 now, which is microbial genetics. There's a lot to this, but hopefully the videos and the summary will help cement what I'm going to go over here in this lecture. There's a few components. There's replication, transcription, translation, and regulation. So effectively 4 items, okay? And it's important to keep those 4 items organized and straight in our minds, because most of the mistakes made by students is confusing one for another. Okay, so what I'm going to do is I'm going to go into my PowerPoint here, and here's really the big picture. Okay. Replication is the manufacturing of DNA from DNA. That's replication. DNA to DNA. So when a cell has to undergo mitosis or meiosis and subsequently cell division, okay, the complement of DNA has to be replicated. So it's a production of DNA from DNA. That's replication. Now, as the cell lives and carries out its function, it's going to need protein, because everything that a cell does is driven by machines called proteins. Okay, so that means that going from DNA to the messenger RNA, that's called transcription. Going from the messenger RNA to the polypeptide, okay, the protein, that is called translation. And then when to turn a gene on or off, that's called regulation. And to cover that, we're going to address the lac and the trip operon. Those are only regulation that takes place in prokaryotic organisms. In us, regulation is significantly more complex. There's a lot going on with us. So I'm going to start with the first part, which is replication. What's replication? Remember, replication is DNA to DNA, a precursor to cell division. Okay. So one of the first things that you need to acknowledge is the fact that this doesn't happen randomly. Okay. There's a rule called Chargaff's rule, named after Professor, a PhD, Chargaff, who realized that there's a relationship between the nucleotides. Okay. So if you look at this PowerPoint, you can see that if you have an adenine, here's a nucleotide adenine, that adenine will always associate with a thymine through 2 hydrogen bonds. Do you see those? Let's make those nice and big so you can see them. There's a hydrogen bond there, and there's a hydrogen bond there. So if you have an A here, it's always going to associate to a T. If you have a T, it's always going to associate with an A. In RNA, the same is true, but the relationship is not between thymine and adenine. The relationship is between uracil and adenine. So in RNA, if you have an A, it's going to associate with a uracil through 2 hydrogen bonds. And if you have a uracil, it's going to associate with an adenine through 2 hydrogen bonds. Going back to both DNA and RNA, okay, if you have a guanine, it's always going to associate with a cytosine through 1, 2, 3 hydrogen bonds. And if you have a cytosine, it's always going to associate with a guanine. What does this mean? This means that if you know one strand of DNA, you'll know the sequence of the complementary strand because of these rules. And again, the Chargaff's rules are G goes with C through 3 hydrogen bonds. In RNA, U goes with A through 2 hydrogen bonds. And in DNA, T goes with A through 2 hydrogen bonds. So here's our DNA, okay? So notice that, again, if you know one strand, you'll know the other strand, because a T is always going to associate with an A G is always going to associate with a C, and an A is always going to associate with a T, and a G is always going to associate with a C, and so on, okay? And here's an explosion of that DNA strand showing the 3 and 2 hydrogen bonds between guanine and cytosine and adenine and thymine, okay? Nothing random. Everything has to happen very specifically. So we're gonna duplicate DNA, okay? We're gonna replicate it. What's going to happen? Well, first of all, the first thing that you need to know is that the energy necessary to make DNA comes from the nucleotides themselves, okay? And in respiration, we said that ATP is energy because of the triphosphate. Well, GTP is energy because of the triphosphate. CTP and ATP are also energy because of that triphosphate. And remember, that phosphate has potential chemical energy because a phosphate is large, is negative, and it wants to dissociate. It wants to move away from each other. It doesn't want to be next to each other. Imagine you have a spring, you have compressed that spring is going to have bountiful amounts of potential energy just waiting to be harnessed to do something, okay? So here it is. Here's our nucleotide triphosphate. And it is this triphosphate that has the energy necessary to incorporate this nucleotide into the lengthening strand. So notice that here we have a strand, okay? And here's the phosphodiester backbone. And so each nucleotide has only one phosphate, but the ones that have not been incorporated yet still have a triphosphate. When the enzyme DNA polymerase associates this guy with this hydroxyl group, that triphosphate turns into a monophosphate and 2 phosphates leave. So again, the energy currency necessary for the polymerization of the DNA and RNA strand are in the nucleotide triphosphates, okay? So that's an important point to grasp. In DNA replication, because at the end of the day, DNA, RNA, proteins, they're all a language, okay? And a language has an orientation. So DNA has an orientation. It has what's called a 5' to 3' orientation, okay? So if we go back to our DNA here, here's our sugar. See my sugar? So a nucleotide consists of 3 things. It consists of a ribose sugar. It contains a nitrogen-containing base, and in DNA, a phosphate group. Well, this sugar the carbons of the sugar are counted 1, 2, 3, 4, and 5. And what you need to understand is that this is a language. So it's just like in English, when we write left to right and top down, because it's a language, in DNA, we always write in a 5' to 3' direction, which means that if we're going to add a nucleotide, we're always going to add it to carbon number 3 and not to carbon number 5. It's always a 5 to 3' direction, okay? So this is important. RNA is the same thing, from a 5' to a 3' direction. So DNA polymerase, the machine responsible for DNA replication, only adds nucleotides in a 5' to 3' direction, okay? Why does this matter? Because DNA is double-stranded, okay? So when you open the DNA to replicate it and form that replication fork, what you find out right away is that there's one side that's a 3', and another side is a 5 prime. Okay, so let's go back to our DNA here. Notice here. See how this is 5 prime and it goes down to 3 prime? And then you look at the other strand, the complementary strand, and that one goes from a 5 prime to a 3 prime in the opposite direction. This is why DNA is said to be anti-parallel, because one strand goes 5 prime and 3 prime in one direction, and the complementary strand goes 5 prime to 3 prime in the opposite direction. Okay? So what happens when you open the replication fork? Well, here's our fork of replication. Fork because it looks like a fork. And replication because that's where we're going to replicate our DNA. And unsurprisingly, one end is a 5 prime and the other end is a 3 prime. Now, what did we just say about DNA polymerase? We said that it only adds nucleotides in a 5 prime to 3 prime direction. That's it. It doesn't work backwards. So when DNA polymerase is replicating, one strand, okay, is adding nucleotides in a 5 prime to 3 prime direction, and that guy just keeps on going. So when the replication fork opens, this DNA polymerase just keeps on chugging along, just keeps on going. It doesn't stop. The other strand, okay, if this is a 3 prime, then this is a 5 prime, and that means that the complementary strand is being polymerized in a 5 prime to 3 prime direction in the opposite direction. So here, before the polymerase can polymerize, it has to wait for the replication fork to open before it can associate with it and form a 5 prime to 3 prime piece of DNA. So this side of the strand where the DNA is polymerized continuously, that is called the leading strand. This other side of the DNA, where it has to wait for the replication fork to open before the DNA polymerase can attach and make another strand of DNA, that's called the lagging strand because it has a tendency to fall behind and therefore lag, okay? So you have, in every strand, in every replication fork, you're gonna have a leading strand and you're gonna have a lagging strand, okay? All right, so those are leading strand and lagging strand. Leading strand is synthesized continuously, nonstop. The lagging strand is synthesized discontinuously in small increments. Okay, the first thing that has to happen is we have to separate the DNA. And that is separated with this beautiful machine called DNA helicase. So this DNA helicase comes in, it produces the replication fork. In other words, it separates the complementary strands of DNA, and it goes right down the replication fork, separating the complementary strands. Now, these two strands, they want to come back together because remember, originally they were being held by hydrogen bonding. And the hydrogen bonding acts like little magnets that want to attract these complementary strands of DNA. So we can't allow that to happen. So what happens is that these stabilizing proteins come together with the DNA and they hold it apart, okay? So the replication takes place. So the DNA helicase separates the complementary strands. Then the stabilizing proteins associates with each strand to keep them separate. And then the next step that happens is that this enzyme, do you see it right there? This enzyme right here, called primase, has to associate with the DNA and produce a little RNA template right there called an RNA primer. And the reason for this is because DNA polymerase cannot polymerize unless it has somewhere to attach to. And that somewhere to attach to is this complementary red RNA primer that has the little 3' here. So once that RNA primer is there, then RNA polymerase goes, oh, okay, we can start. And then it starts polymerizing. You see that? So no surprise, in the leading strand, there's only going to be one RNA primer. However, in the lagging strand, okay, whoops, let's go back up. In the lagging strand, here's our lagging strand. That primase associates, it makes a little RNA primer. Then the DNA polymerase attaches, it forms DNA, okay? And then the replication fork opens again, the primase attaches, makes another RNA primer. Then the DNA polymerase attaches, and it polymerizes it a little bit. And then the replication fork opens again, and another primase attaches over here, and another primer gets made. And then DNA polymerase attaches, and it makes another little guy right here, okay? So if you're noticing, on the lagging strand, we don't have one continuous piece of DNA. We have a whole bunch of small little pieces. Each little piece is called an Okazaki fragment. It's named after the Japanese scientists that discovered them, okay? And so you have, in this illustration, you have 1, 2, 3 Okazaki fragments. And once they're formed, when you need more help, okay? So notice that the guy synthesizing the DNA is DNA polymerase 3, but once this DNA reaches the next primer, this guy comes off and a different machine needs to come in. The name of that machine is DNA polymerase 1. So a DNA polymerase 1 now attaches, it removes the RNA primer, okay? That's called the exonuclease activity. You don't need to remember it. But it removes the primer, it replaces it with DNA. So it replaces RNA with DNA. Then it comes off, and one more machine comes on. That machine is called DNA ligase. And that DNA ligase attaches here, and it makes a covalent bond between these two nucleotides. And now we have a complete strand. A lot of work, isn't it? Okay. So to recap, first guy that comes in is DNA helicase that causes a replication fork to open. Then you have these stabilizing proteins that maintain the DNA separate. Then primase associates. It makes an RNA primer. Then DNA polymerase 3 associates to the 3' end of the RNA primer. On the leading strand, it continuously synthesizes the daughter strand. But on the lagging strand, that DNA polymerase only synthesizes a small fragment of DNA called the Okazaki fragment, and it stops when it abuts the RNA primer of the next Okazaki fragment. Once it abuts it, it comes off, and then DNA polymerase 1 associates. It cuts out the RNA primer and replaces it with DNA. And then once that's done, it comes off, and then DNA ligase comes in and forms that last covalent bond between the two Okazaki fragments. Quite a bit of work, huh? So that is DNA replication. I encourage that you take a minute break. I know I'm going to, and then we're going to move on to transcription. Okay. Okay, so we just finished replication. Now we're going to move over to transcription. Okay. Transcription. Where are we going from? We're going from DNA to messenger RNA. That's what we're doing. As your author states, there is 3 steps to it. There's initiation, elongation, and termination. So let's start at the beginning. Here's initiation, okay? So this is DNA. Now remember, we're carrying out transcription. So in replication, the machine was DNA polymerase because we're turning DNA into DNA. This time, we're carrying out transcription. We're going from DNA to messenger RNA. So the machine that we're going to use is called RNA polymerase because it's polymerizing the message, okay? So here's our beautiful DNA strand right here, okay? And we have initiation. And your author very nicely states that the way that this works is that the RNA polymerase associates non-specifically to the DNA strand. So you have the DNA strand, and then RNA polymerase comes up, and it just attaches, and it just goes down the DNA strand, okay? And it looks like it's looking for something, and it is looking for something. What it's looking for is a specific sequence of DNA nucleotides called a promoter, okay? When the RNA polymerase finds this promoter sequence, it attaches itself even more tightly to the DNA strand, causing the DNA strand to open up and make a bubble, okay? In prokaryotes, there's a protein called a sigma factor that facilitates this, and it makes the RNA polymerase attached even stronger to this promoter region. Once this has happened, okay, we have a bubble, and now that RNA polymerase will start incorporating RNA nucleotides. Remember, the RNA nucleotides bring their own energy because they're going to be RNA nucleotide triphosphates, okay? And is going to be incorporating it into the new messenger RNA. In what direction? It should not be a surprise. It's always going to be in a 5' to 3' direction. So the new nucleotides are going to be incorporated into the 3' end of that message. See that 3' right there? That's where the new nucleotides are going in. So here's the 3' end, and here's the new nucleotides. And notice also that they're being incorporated complementary. Let me make this even, I always have trouble. I always go backward with this thing. They're being complemented, they're being added with a complementary nucleotide on the template DNA strand. So here's a DNA strand. If you have an A, it associates with uracil from RNA. Remember Chargaff's rules? The same Chargaff rules that work in DNA are similar to the Chargaff rules that work in RNA. Here in RNA, U goes with A, G goes with C, and you can see that as you see the template strand, there's going to be a G with a C, there's going to be a T with an A, there's going to be an A with a U, there's going to be a C with a G. These are the same Chargaff rules that we studied just a little bit ago. So the messenger RNA is being synthesized complementary to the DNA template strand. That makes sense, right? And this work is being energized by the triphosphates of the nucleic acid and is being carried out by this beautiful protein called RNA polymerase. So this event where additional nucleotides are being added to the nascent messenger RNA strand, that's called elongation, okay? And that's going to happen until we reach some sequence of DNA nucleotides called a terminator where the RNA polymerase is going to slow down, okay? So we're heading in that direction, we're headed towards the terminator, and once we get to the terminator, we have to stop, okay? So here's the DNA The RNA polymerase has reached the terminator and it gets to stop. There's two ways that it can do it, okay? It can do what's called a self-termination. So once it's gotten to that area, what happens is it runs into what's called a GC-rich area. So if you remember from Chargaff's rules, G and C come together with 3 hydrogen bonds, and U and A come together with only 2 hydrogen bonds. Well, which one sticks together more tightly? Well, the answer is the GC, because it has 3 instead of 2 hydrogen bonds, so they have a tendency to stick together. GC has a tendency to stick together more tightly than UA does, okay? So the great architect, what he did is, on some instances, there's a G-rich area followed by a C-rich area. And what happens is that when this message gets synthesized, the G and the C come together, and they literally do this, swap. They just come together really fast. And when it does that, it knocks the message off of the DNA, and it removes the RNA polymerase off of it. So this is called self-termination, okay, or protein-independent termination, or it can use protein-dependent termination. In this case, it uses a protein called Rho protein, okay? You can see that we've reached the same terminator region, that orange region there. It's time for the RNA polymerase to dissociate from the DNA. And so there's this little protein that runs up the mRNA, and when it gets to this terminator region, it gets between the messenger RNA and the DNA template strand. It pulls them apart, and it has the same effect. It pulls the message off of the DNA template, and consequently pulls the RNA polymerase off of it. So this is self-termination or protein-independent termination, and this one is Rho-dependent or protein-dependent termination. Okay. Now, don't you enjoy this? I mean, isn't it beautiful? Remember the choreography in DNA? You have helicase, you have strand proteins, you have polymerase 3, you have polymerase 1, you have, I mean, oh my goodness, what a mess. And then you go to transcription from DNA to RNA, and one guy does it all. One little guy, okay? Now, one of the things that allows it to be one little guy is that in DNA replication, you have to end up with 2 strands of DNA to 2 strands of DNA. But in transcription, you have 2 strands of DNA, but one of them is a template, and you have to end up with a single strand of messenger RNA. That simple event makes everything easier, and as a consequence, you end up using a single protein, messenger RNA polymerase, instead of 5 or 6 different proteins like in DNA synthesis. Very incredible stuff. So we're going to stop here, and I'm going to continue with the other 2 parts in just a second.